Hello everyone. I welcome you all to Biology Lessons with Kalyani. I hope you all are doing very good. Today we are going to start with the topic Nucleotides, another chapter Biomolecules, CBSE Class 11 Biology. And let's look at what do we have in our topic. So this is the screenshot of the NCERT textbook. I have highlighted the important points. So all these points we will be covering in detail today. So let's begin with the topic. We all know that the nucleic acids, we all know that the living beings, all the living organisms, they consist of nucleic acid. So what does this nucleic acid consist of? This nucleic acid consists of the genetic information Now, nucleic acid can be of two types. It can be either DNA or it can be RNA. So, DNA is nothing but deoxyribonucleic acid and RNA is ribonucleic acid. Now, we can call this DNA and RNA as either nucleic acid or we can alternatively also tell them polynucleotide. Now, if you see, polynucleotide has the word poly in it and nucleotide in it. It means that DNA and RNA or the nucleic acid are polymers of nucleotide. So, nucleic acids are essentially the polymers of nucleotides. Alternatively, we can also tell that many nucleotides join together and form a polynucleotide or a nucleic acid. Or in other words, nucleotide is the monomer for nucleic acids. Like amino acid is the monomer for protein, nucleotide is the monomer for polynucleotide or nucleic acid. Hence nucleotides are the building blocks of nucleic acid. I hope this introduction part is clear for you all. Let's move on to the next slide. If we talk about the components of nucleotide has three main components. How many components does a nucleotide has? Nucleotide has three main components. What are they? Number one is the pentose sugar. Number two is the nitrogenous base. And number three is the phosphate group. Clear? So we can write nucleotide is equal to pentose sugar plus nitrogenous group plus phosphate. So when the three combine together, they form nucleotide. Now when both pentose sugar and nitrogenous base combine together, we call it nucleoside. So what is nucleoside? Nucleoside is is pentose sugar plus nitrogenous base. Okay, this is nitrogenous base. Is it clear? So we can also write it as nucleotide is when nucleoside combines with a phosphate group, or in other words we can tell that nucleotides are phosphoesters of nucleoside. Very important. Nucleotides are phosphoesters of nucleoside. I hope this part is clear. Nucleotide has three parts: pentose sugar, nitrogenous base, phosphate. Pentose sugar, when it combines with nitrogenous base, it is called as nucleoside. So nucleotide is nucleoside plus phosphate, or nucleotide is phosphoesters of nucleoside. Okay, let's now look into all the different components in detail. So let's look at the first component here, which is pentose sugar. As the name suggests, pentose sugar will consist of 5 carbon structure. Okay. Now the nucleotides, they can have two types of pentose sugar. Two types of pentose sugar. If we consider RNA, then the pentose sugar in it is ribose. Now, if we consider DNA, then the pentose sugar which is present in it is deoxyribose. Okay. So, RNA will have ribose and DNA will have deoxyribose. Next thing is the pentose sugar has 5 carbons. Now, this is present always in DNA and RNA in the form of its cyclic structure. This 5 carbon cyclic structure is called as furan structure or furanose structure. And it is also the beta type. That's why we can also call it the ribose and deoxyribose exist as beta furanose structure. Now, what is this beta? I will explain in the diagram. Okay. Now, let's learn how to make ribose structure. To make ribose, first start by placing an oxygen atom. Then make a 5 carbon ring structure. Now, there are only 4 carbons in the chain. Now, the 5th carbon will be always present outside the ring structure. So, this point you have to remember. 4 carbon atoms are present in the ring structure and the 5th one always is outside the ring structure. Okay. Next, we will put the other groups. So we will just add HOH, HOH, and the fifth carbon is CH2OH. Okay. So now let's look at the numbering of the carbon atoms. For numbering, we always start from the right of the oxygen atom. See, this is the 1 prime, this is 2 prime, 3 prime, 4 prime, and 5 prime. The fifth carbon is always outside the ring structure. Now, if you mark the fifth carbon, the hydroxyl group present in the fifth carbon and the hydroxyl group present in the 1 prime carbon, they are in the same direction or pointed upwards. Hence, this structure is called as beta type structure. If the OH group of fifth carbon is pointed upward while the 1 prime hydroxyl group is pointed downwards, that type of structure is called as alpha type structure. So here we have both pointed upwards. So this type of structure is called as beta furanose structure. Okay, I hope that point is clear. So both OH groups of fifth carbon and 1 prime carbon pointed upwards, then that type of structure is called as beta furanose type structure. Why this term beta is important? Because when it makes the bond, there beta glycosidic bond will come. That's why it is important to know why we use the term beta here. I hope that point is clear. Next important point is, what is deoxyribose? Now see, in this structure, if we take out the oxygen from the 2 prime carbon, then if we remove oxygen from 2 prime carbon, then deoxyribose will be formed. Deoxyribose. Deoxy means oxygen is removed. From where? From the 2 prime carbon of the ribose. So how does the structure becomes? Instead of OH, there will be just one H present. So rest all it remains the same. So start by putting OH, H. Rest all I will put first H, OH, H, CH2, OH. Now what happens here? Here in the 2 prime there would be H and H. Instead of OH, only H will be present. Instead of OH, H will be present. So this is deoxyribose, which is the foundational component of DNA. Foundational pentose sugar for DNA and ribose for RNA. I hope this is clear. Let's now talk about the second component of nucleotides, that is nitrogenous base. Now, nitrogenous base are a group of heterocyclic compounds, heterocyclic organic compounds. So, what is the meaning of heterocyclic organic compound? It means that it has other atoms apart from carbon in its ring structure, such as nitrogen. Now, these nitrogenous bases, they are derivatives of purines, and pyrimidines. Okay. Now, the structure of purines is essentially a 9-membered double ring structure. Okay. What is the meaning of this? That means that purines comprise of a 6-membered ring structure which is similar to pyrimidine plus imidazole ring structure. So purine consists of a 6-membered ring structure plus imidazole ring structure. And what about pyrimidines? Pyrimidines, they consist of a 6-membered ring structure. Okay. So you can remember like this, that even though the name purine is small, they have a bigger ring structure. So they have 2 rings attached or fused in them, while pyrimidine have a single 6-membered ring structure. Right? Now, what comes under purine? Under purine, you have adenine and guanine. Under pyrimidine, you have cytosine, uracil, and thymine. Okay, so how do you remember this? You can remember this as big purine got some apples and grapes. Apple is nothing but adenine and grapes is guanine. And small pyrimidine, cut it. Cut is C cytosine, U is uracil, P is thymine. I will give the mnemonics in the I will give a link for that and you can refer to it later. Okay. Now, if we see the purine, it remains the same in both DNA and RNA. So, in both DNA and RNA, purine is same. Okay. So, what differs? Pyrimidine is different in DNA and RNA. In RNA, only uracil is present, while in DNA, thymine is present. So what's common pyrimidine in case of both DNA and RNA? Cytosine is the common pyrimidine. Is that clear? So nitrogenous bases are of 2 types, purine and pyrimidine. Purine consists of A and G, which is nothing but adenine and guanine, and pyrimidine consists of cytosine, uracil, and thymine. Now, the purine remains same in both the DNA and RNA, while the cytosine is the only common pyrimidine in both DNA and RNA structures. RNA has uracil while DNA has thymine in their structure, right? I hope this is a basic about nitrogenous base. After this, we will be looking a little more into their structure, how to name the nitrogenous bases, and so on. Okay. Let's see the next slide. Let us now look into the structure of purines in a little bit detail. Okay. So purines we discussed, they consist of adenine and guanine. Now, if we look into the structure of purines, they have a fused structure in which there is a six-membered ring plus imidazole ring. That's the one thing you have to remember about the structure. So they have a fused double ring structure. Now, next point is, where do we put, because there are nitrogen atoms, right? There are nitrogen atoms. Next point obviously comes, where do we put the nitrogen atoms? So the nitrogen atoms you have to put in all odd numbers except number 5. Okay, so what does that essentially mean? That essentially means you put it in number 1, number 3, don't put it at 5, then put it at 7 and 9, right? So this is second thing. So let's try to make the structure first. Basic structure of purines. So there would be a six-membered ring structure. Fused to it, there would be imidazole ring structure. So let's put the double bonds here. This is the core structure. Now let us put the nitrogen. Always for the numbering thing, you always start from top left nitrogen. Okay, and let us put the nitrogen first. So this is the number 1 position. Put 1 nitrogen. 2 we don't have. 3 we have. 4 we don't have. 5 is the odd position in which we don't have the nitrogen atom. Then 6 doesn't have, and this is 7. 8 doesn't have. 9 has. So we put the nitrogen atom. Next is the counting. So if we consider the 6-ring structure, the counting is always opposite to how the clock moves, or anti-clockwise. And for the 5-ring, it is clockwise. For the 6-ring, it is anti-clockwise. And for the imidazole, it is clockwise. This is second point you have to remember. Okay, so now let us put the numbers. Okay, so this is carbon number 1, 2, 3, 4, 5, 6, and then you have 7, 8 and 9. Clear about the numbering? I hope this would be clear. Now we know everything about the purine structure. Just to this core structure, we would be making some modification and we'll be making adenine and guanine. So let's first make the core structure for adenine. Okay, we put one nitrogen here, one nitrogen here. And another thing also you can remember, we put the functional groups in the even number because odd number are having the nitrogen. And the type of functional group we are going to put is always going to be amine, mostly amine and carbonyl group. Okay, so in this case for adenine, you would be putting one amine group here. So let's add the double bonds. Okay, so let's satisfy the valency. Okay, so this is the structure for adenine. Adenine structure, I hope it is clear, very easy to remember. So this has the side chain which you have to attach is 6 amino. Just remember it and remember the overall structure of purine, and you can easily make the structure of adenine. Now let's try to make the structure of guanine. So make the base structure, 6-ring structure and imidazole structure. Put one double bond here. Okay, nitrogen atoms. We put everything. So why I didn't put the double bonds? Because in the core structure, it may be modified a little bit to make other structures. So for guanine, what happens? The amine group, which was attached to the 6th carbon, it comes to the number 2 carbon. Okay, and in the 6th place, there would be carbonyl group. Okay, so it becomes guanine becomes 2 amino 6 carbonyl. So you can remember as 26 AC. Okay, AC air conditioner, we all have to 26 AC, 2 amino 6 carbonyl. Is the structure of purine very clear? I hope you should be very easily able to make the structure of purine. Just remember the mnemonic and then you can easily draw it and the basic fundamentals which you require for making the structure. So let's now see pyrimidine structure. So pyrimidines, they essentially are 6-membered ring structure. 6-membered ring structure. So it consists of cytosine, uracil and thymine. So cytosine is common in all. Uracil is only found in RNA and thymine is found in DNA. Clear? So here I'll first try to make the structure of uracil. Before that, let us now know what is the core structure of pyrimidine. So pyrimidine, it essentially has a 6-membered structure and it also has the nitrogen atoms in all odd except number 5. So it is a 6-membered ring, so it cannot have nitrogen atoms at 7th and 9th position. So nitrogen atoms, it will be present in position 1 and position 3. And the numbering always starts from the base nitrogen and it is always in the numbering is always in the clockwise direction. Okay, so this is number 1, 2, 3, 4, 5 and 6. Clockwise counting, and it is always starts from base nitrogen. This is the fundamental you need to remember. The rest all you can easily make it. So let's start by making the structure of uracil first. Okay, that's very easy. Make the core structure. Just put one double bond here. Don't put anything else yet. Put the base nitrogen atom here. 1, 2, 3. Third nitrogen atom in the third position. You have put it. Okay. Now uracil has two carbonyl groups attached to them. I told the functional groups are always attached to your even numbers. Why? Because odd numbers are occupied. Nitrogen atom is there. So, two carbonyl groups are attached to the second and the fourth position. Now, because the carbonyl bond is made, the double bonds are not present here. Okay. In the core structure, you had the double bond in this position and this position. They are absent in uracil. Okay. Rest, you just satisfy the valence. So, NH. Okay. So, this is the structure for NH. This is the structure for uracil. Numbering, let's start. 1, 2, 3, 4, 5, 6. So you have to remember 2, 4 carbonyl you are adding. Okay. 24 carbonyl. So 24 carbonyl. Write that, you remember. Okay. So uracil structure done. If uracil is done, you can do all rest. Thymine structure is exactly similar to uracil, only in the fifth position they have extra methyl group. So you can write as thymine is equal to 5 methyl uracil. So what do you do here? Make the core structure. Don't put the other double bonds yet. Put the nitrogen atoms. Then put the carbonyl group and at the second and the fourth position, and in the fifth position attach one methyl group. Finished. This is the structure for thymine. Now check with the numbering. Start from the number 1 position here. Go clockwise. 1, 2, 3, 4, 5, 6. That's it. So simple. This is the structure for thymine. You can easily remember it. Uracil, thymine, and let's now make cytosine, which is common in both DNA and RNA. Now let us make the core structure. Okay. Let us now make the core structure. So here what happens? We can make the double bond here, nitrogen atom, here one nitrogen atom. After putting that, we do not change the carbonyl group at second position. This remains intact. What changes? Here just you put the amine group. So to satisfy valency, you can put one C double bond N or imine bond here. Okay, that's it. The structure of cytosine is done. So let's number it. 1, 2, 3, 4, 5, 6. So essentially, cytosine structure, in the second position you have the carbonyl group just like the uracil. Only in the fourth position you put the amino group. So remember it like this. 4 amino, 2 carbonyl. Okay. Okay. In this way 42 AC. This way you can easily remember the structure of pyrimidine. Just remember the base rule first and start by remembering the structure of uracil, and then you can automatically make the rest of the structure. If you know uracil, you can easily tell the structure for thymine also, because thymine is nothing but 5-methyl uracil. Clear? Let us now look at the final component of nucleotide, that is the phosphate group. Now, this is phosphorus atom, double bond O, 3 OH groups are attached to it. This is the phosphate group or orthophosphoric acid. Now, this has a tendency to lose protons. Now, this will interact with the OH minus of the pentose sugar. In this, then there would be an ester bond formed. Alongside, water will be released, and the type of the ester bond will be called as phospho-ester bond. We'll be looking into a little more detail to the detailed phospho-ester bond, how it is formed in the coming slides. Okay, for now, you can just remember this is the structure of the phosphate group. Okay. Let's now see how the nucleosides and nucleotides are named. Now, we know that RNA, let us look at RNA first. Now, RNA consists of A, U, G, C. Okay, A and G are the purines, while U and C are the pyrimidines. So now, this is adenine, this is uracil, this is guanine, this is cytosine. When this will react with ribose, nucleoside will be formed. That will be called as adenozyme. Okay, adenozyme. And uracil, when it will react with ribose, it will be called as uridine. And guanine, guanozyme. And cytosine, cytidine. If you see, for uracil and cytosine, which is the pyrimidine, D is used. Dine, okay? Adenine, adenozyme. So these all are the nucleosides. Now, if it will react with the phosphate group, then respective nucleotides will be formed. So how can we name the nucleotides? You can say adenylic acid. Otherwise, we can also say adenylate. Uridine plus phosphate makes uridylic acid or uridylate. Similarly, guanosine plus phosphate makes guanylic acid or guanylate. Cytidine plus phosphate makes cytidyllic acid or cytidine. dilate. Okay. In case of DNA, if we see, we will have A, T, G, and C. Okay. And the sugar will be deoxyribose. This is mentioned. This is the nucleotide. Okay. Deoxyribose. So here just we need to write the deoxyadenosine. Deoxyadenosine, that is the nucleoside. Similarly, deoxythymidine, deoxyguanosine, okay, deoxycytidine, okay, and once it will react with phosphate, deoxyadenylate or adenylate acid. I'm just writing one here. You already can write deoxyadenylate acid. Okay. Deoxyadenylate acid. Similarly, you can make for others. Okay. This becomes deoxythymidylate. Deoxyguanylate. Deoxycytidylate. Okay. I hope this nomenclature part is clear. This is the side and this is the nucleotide. Interesting, right? Let's see the final part, that is how the bonds are formed and how the nitrogen base is reacting, where it is reacting, how the phosphate group is reacting, how it is reacting. Let's see in the next slide. Let us now look at what are the bonds present in nucleotide. We will just have an overview here. Detail we will take another lecture for the different types of bond present in nucleotide. But let's have an overview first. So we know that nucleotide, it has a pentose sugar, then to it is attached nitrogenous base, and we then attach a phosphate group. I hope this part is clear. So if we consider, let's take here ribose sugar. Okay, so this is OH, this is H, and this is your H, OH, this is H, OH, this is CH2OH, and this is H. Okay, so this is ribose. Now the 1' of ribose will always interact with the 1' of ribose will always interact with the nitrogenous base, and the type of bond formed is beta glycosidic bond. Now why beta? Because we use beta ribose here, right? Now This glycosidic bond is formed in case of, if you consider urine, it is made the N9. Okay, the ninth nitrogen atom. And case of the pyrimidines, N1. Okay, so this is how the glycosidic bond. So where will it exactly attach? This H will be interacting with the, I'll just take example of 89 here. What is the structure of 89? Structure of 89 is have a double bond structure. Okay, an imidazole ring. Let's make that imidazole ring. Okay, then nitrogen atoms, amino group. So this is your number 9, which will, this is number 9. This nitrogen form the bond with your, this nitrogen form the bond with OH of the ribose sugar. So this H and OH interacting with each other, and there will be a loss of water molecule and beta glycosidic bond is formed. If you take an example of, so this is your N9 for purines. Okay, if you take example of pyrimidine, let's take example of uracil here. Which is nothing but, this is uracil. So here the carbon number 1 will be forming the glycosidic bond. Okay, again this will react with the ribose sugar OH and at the 1' and glycosidic bond will be formed. So 9th nitrogen for purine and 1st nitrogen for pyrimidine. Remember this now. We will in detail study it little later. Okay. Now let's check what the phosphate group is doing. Where is it getting attached? Okay. So this is your carbon number 5 for the ribose group. Now let's place the phosphate group here. This is a phosphate group. Now we know phosphate group has a tendency to lose what? Protons. So now this will lose a proton, OH from the ribose sugar, loss of one water molecule, and what is the type of bond formed? Ester bond in common and phosphoester in specific. So where is phosphoester bond formed? C prime reacts with the phosphate group, or the 5th carbon atom reacts with the phosphate group and forms the phosphoester bond. Okay, this is very, very, very important. Okay, C prime, C first prime of the ribose interacts with the bases to form the beta glycosidic bond. 9th nitrogen for purine and 1st for the pyrimidine, N9 purine, N1 pyrimidine, and phosphoester bond is formed between the 5th carbon atom of the ribose to the phosphate group. Okay, So let us write ribose here also, C1 ribose, okay. I hope this part is very clear. With this, we end this lecture session. I hope you got the concepts about what are nucleotides, what are the different components in nucleotides, how to make the structure, how to do the nomenclature, what's the difference between nucleoside and nucleotide, how to write them, what are the different bonds present in nucleotides. So I hope you are clear about all these things. Now this chapter is very important because it is the foundational chapter for understanding the structure of DNA, understanding molecular basis of inheritance. So I'm very sure that you would have definitely enjoyed the session. Please hit the like button and share and comment and subscribe to the channel. Thank you everyone. Have a very good day.